Good afternoon, everyone. My name is Hannah Fuller, and I'm a media officer with the National Academies of Sciences, Engineering, and Medicine. Thank you for joining us this afternoon for a webinar on the report that was just released this morning titled Charting a Future for Sequencing RNA and its Modifications, a New Era for Biology and Medicine. You can now download a copy of the report and other supporting materials at www.nap.edu. A recording of this webinar will be available on our website immediately after it is over. For those of you not familiar with the U.S. National Academies of Sciences, Engineering, and Medicine, we are private, nonprofit institutions that provide independent, objective analysis and advice to the U.S. to solve complex problems and inform public policy decisions related to science, technology, and medicine. For each requested study, panel members are chosen for their expertise and experience and serve pro bono to carry out the study's statement of task. The reports that result from the study represent the consensus view of the committee and must undergo external peer review before they are released, as did this report. I have the members of the committee who wrote the report here with me today, but before we introduce them, I want to go over a few reminders. Please note that this webinar is scheduled to last 1 hour. We'll start off with a presentation summarizing the report by the committee members, and then we'll open it up to any questions you may have. To ask a question, just click Q&A at the bottom of your screen and type your question. You can submit a question at any time during the presentation, and we'll get to as many as we can at the end. Now I'd like to turn it over to Dr. Victor Zhao, President of the National Academy of Medicine, for some opening remarks to frame the importance of this report. To you, Victor.
Thank you so much, and good afternoon, everyone. I'm so excited that we're releasing this report, and thank you for joining us today. The report is called Charting the Future for Sequencing RNA and its Modification, a New Era for Biology and Medicine. You know, this report highlights an important frontier in biology research that can truly transform medicine. In 1988, the National Academies released a seminal report on mapping and sequencing the human genome. It was groundbreaking, as you know, because it paved the way for this massive international effort in human genome sequencing and subsequently the genomic revolution. In my opinion, today's report RNA has the same transformative potential. So over the past several decades, as a result of the Human Genome Project I mentioned, we've witnessed a remarkable trend towards precision medicine, driven by impressive advances in genomic technology, data, and of course, application to health and diseases. But we're also learning through great research. Some of the members of the committee, their work, and many others, there's a lot more story than the genetic code. In fact, you'll hear today, RNA and the modification it acquires over the course of its life cycle, called RNA modifications, are pivotal players in nearly all biologic processes, and that dysregulation has been implicated in numerous human disease and disorders. Indeed, these modifications offer a wealth of opportunities for understanding, addressing diseases at a much deeper level, and offering unique solutions and novel therapies for new and existing health challenges. So understanding RNA modification and harnessing its knowledge has immense potential, as you hear, but not only for human health and medicine, but also shaping all living systems and the products and technologies that stem from them, including those vital for the bioeconomy, such as agriculture, food, and many others. This consent study provides a roadmap for RNA sequencing research, which, as you hear, will require significant commitment and investment in new technologies and capabilities. But I think the excitement about this is that it will open up so many opportunities for great research. At the end of the day, that information can help human beings and many other living systems. So when I reflect on similar undertakings in the past, like the Human Genome Project, I think this work on RNA modification sequencing will require extensive collaboration among scientists across disciplines across organizations and certainly internationally. And it will require, in my opinion, strategic public-private partnerships. So, we really are at a very exciting time. Truly understanding the complexity of biology is about weaving together the intricate tapestry of multi-omics data to paint a comprehensive picture of biology and ultimately enhancing understanding of human disease, agriculture, environment, bio-based economy, and defense. So before I turn over to the chair of the meeting, I'd like to thank a number of people. Now, first of all, Brenda Bass and Tekjip Ha, the co-chairs of planning committee, and of course, their entire committee, some of them are here today. They have just done outstanding work. But it couldn't be done without major staff involvement and leadership. And I want to call out Tricia Tucholsky for her work in supporting and shepherding this project, because under the guidance of Kavita Berger, they are just the best. I'm so pleased to be here to be discussing this report due to all your hard work over the last few months. And I'm so happy to be here to discuss the report publicly now. Finally, you're going to hear the findings. But I particularly want to thank the generous support of Warren Alpert Foundation and NIH, especially the Warren Alpert Foundation, because they saw the importance of potential funding of the study, and they have provided us with unwavering support and funding. And I just want to thank them very much. So with that, let me turn it over. Oh, wait a minute. I have a few more things to say. I want to invite you today to explore building the repertoire of RNA modifications in the pursuit of understanding the multi-omics research as potential to transform healthcare, improve lives. So thanks once again for your participation. Look forward to a really insightful discussion. And now let's embark this journey beyond the genome. Thank you, and back to you.
Thank you, Dr. Zhao. I'll now pass it to Dr. Tricia Tucholsky, a program officer in the Division on Earth and Life Studies at the National Academies, to take us from here.
Thank you so much, Hannah and Victor, and thank you for all joining us for the public release of our new report, Charting a Future for Sequencing RNA and Its Modifications, a New Era for Biology and Medicine. My name is Tricia Tucholsky. I'm a program officer with the Board on Life Sciences at the National Academies and the director of the study that resulted in this report. And before we begin, I just want to pile on my acknowledgements to the people that made this report possible. First, I'd really like to extend a special thank you to the members of the study committee for their service. It has been an absolute pleasure and privilege to work with this dedicated and knowledgeable group of experts. The report underwent a rigorous review process, so we would also just like to thank our volunteers who served as reviewers. I'd also like to express my gratitude to the National Academies project team and our colleagues across the academies for helping us in so many ways throughout the study. I also want to acknowledge Stephen Moss, who directed the study up until August of last year and set us up for great success. And lastly, I really want to thank our study sponsors, the Warren Alpert Foundation and NIH, specifically National Institute of Environmental Health Sciences and the National Human Genome Research Institute, for their generous support of the study. And without further delay, I will pass it over to our committee co-chairs, Dr. Brenda Bass of University of Utah Medical School and TJ Ha of Boston's Children's Hospital and Harvard Medical School, to give you an overview of the report and highlight some of the key findings and recommendations. So Brenda, TJ, over to you.
Thank you, Victor and Tricia.
So are you seeing the correct screen? Oh, not yet. Okay, there we go. How about that?
Presenter view, Brenda.
Are things okay on that end, Tricia?
We still see the presenter view.
Oh, thank you. I didn't hear you.
There we go.
Okay.
Sorry.
Okay, it's good.
All right. Good afternoon, everyone, and thank you for joining us. We are truly excited to be here today to tell you about our new report titled Charting a Future for Sequencing RNA and its modifications, a new era for biology and medicine. I am Tekchip Ha, a study co-chair. I will first tell you how the study was performed, and then my co-chair Brenda Bass will tell you what's in the report.
Okay, let's go.
This report was just released today at 11 a.m. Eastern Time and can be downloaded at the link on the screen. You can also scan the QR code. The report was authored by the Consensus Study Committee toward sequencing and mapping of RNA modifications. The study was jointly supported by the Warren Alpert Foundation and the National Institutes of Health, specifically the National Human Genome Resource Institute and the National Institute of Environmental Health Sciences. It is a collaboration between two boards at the National Academies, the Board on Life Sciences and the Board on Health Sciences Policy. We have a few people to thank before we get started. I was really privileged to chair the committee with Brenda Bass. I would also like to thank and acknowledge the excellent members of the study committee, many of whom are here today, and a few will be participating in our Q&A panel at the end of our presentation. We are fortunate to have had a really amazing and outstanding committee of 16 experts from academia, industry, and government with diverse and complementary expertise. We'd also like to thank the staff at the National Academies for their work on the study. The amazingly capable study directors, initially Stephen Moss, and then Trisha Tchaikovsky, kept us on track and on time, and all of the staff members were incredible in helping us write this report. We send a special thanks to Janet Iwasa and Rachel Torres for designing clear and accurate scientific figures and graphics. The report underwent rigorous peer review by 12 experts, shown here, with expertise spanning the RNA modifications field, including those with knowledge of different sequencing methods, policy, and workforce. We thank them for their helpful reviews that allow us to further enhance the report. We also would like to thank the review coordinator, Paul Alquist, and monitor, Dan Blazer, for overseeing the review process and providing additional support and guidance. The central goal of this report was to provide a roadmap to guide the development of tools, technologies, and infrastructure that are needed to realize the capability of sequencing any RNA, including all of its modifications, in a robust and accessible way. To develop this roadmap, the committee was guided by a statement of task that specifies areas to focus on, including scientific, technical, computational, policy, infrastructure, and workforce needs. The committee also carefully considered each component.
Are you still seeing, did you not see this anymore?
Now we are seeing your desktop.
Okay, well, let's see what happened there. Sorry about that. Okay, let's share screen again and see what happens. And I saw that go for it, but you're doing a great job so far, TJ. And
did you want me to share them on my end?
I think we're almost there. I don't know why that happened. Is it back and do we have the right display or not?
We don't see
Not
them.
yet.
Okay, you don't see it yet. Okay, how about that?
No.
No.
Trish, can we do it on our end?
Yep, I can. Yep, absolutely.
Brenda, why don't you let us do it? And you can just say next slide.
Okay, okay. Let me
There you go.
Okay, let me get back to this slide. So to develop this roadmap, the committee was guided by a statement of task that specified areas to focus on, including scientific, technical, computational, policy, infrastructure, and workforce needs. The committee carefully considered each component and designed a series of information gathering activities to collect a diverse set of perspectives related to each topical area. Next slide. The committee began its information gathering process by reviewing previous workshops and literature related to the field of RNA modifications, including a recording and report from an NIH workshop that was held in May 2022, capturing RNA sequence and transcript diversity, from technology innovation to clinical application. Then the committee held a 1.5-day workshop in March 2023 that covered topics such as the current state of RNA modifications technologies and standards, the application of RNA modifications to health and medicine, and societal implications of RNA modifications research. The proceedings in brief for this workshop is published and is freely available online. In addition to this workshop, the committee held several other information gathering sessions and meetings with experts. Over the course of the study, the committee heard from approximately 50 different speakers, panelists, and discussion participants. The committee also held an ideation challenge in June 2023 to encourage collaboration and spur innovation. This event brought together 38 researchers to brainstorm about innovative ways to address challenges and technical hurdles. And 3 of the 7 working groups there were asked to expand on their ideas and wrote commission reports that are in the appendices of the report. The committee also referenced, can you go back? Thank you. The committee also referenced past National Academies reports that spurred important scientific initiatives in life sciences, specifically the report from from 1988 titled Mapping and Sequencing the Human Genome that preceded the Human Genome Project, and a more recent report titled Transforming Glycoscience, a Roadmap for the Future, which preceded the NIH Common Fund's Glycoscience Program. Next slide. The report is organized into 6 chapters listed here, in addition to a summary and appendices. The summary is a great place to start because it highlights all major conclusions and recommendations. Chapter 1 provides an introduction and brief history of the RNA modifications field. Chapter 2 covers the importance and impacts of RNA modifications. And Chapter 3 examines the current state of tools and technologies for identifying, quantifying, and sequencing RNA modifications. It also lays out gaps and challenges that are not addressed by the current technologies and explores future areas of technology development that would impact the RNA modifications field. Chapter 4 then takes an in-depth look at the current state of RNA modification standards and databases, which will be key in advancing research and technology development efforts. Chapter 5 explores possibilities for driving innovation in the field of RNA modifications, including workforce needs. And the final chapter, Chapter 6, synthesizes information from prior chapters into a set of key conclusions and recommendations for driving the field forward. It also provides a set of roadmaps that we hope will be helpful, including interim deliverables and timelines. Chapter 6 also offers guidelines for when different goals should be accomplished. And now I'll pass it over to my co-chair, Brenda Bass, to tell you about why we are so excited about the prospect of sequencing RNA and all of its modifications. Brenda.
Thank you, TJ. So now we want to talk about our favorite molecule, RNA. And because I don't have a pointer, I'm going to try to bring you into my head. So most people are more familiar with DNA. And on this slide, you see two strands of DNA, the black. Oh, thank you. Maybe that's NAM. The black and the gray. And this molecule DNA encodes our genes. It safely stores the blueprint for life. DNA does not travel within a cell, but stays in one place, called a nucleus in human cells. Its information is passed to different parts of the cell by a molecule called RNA. RNA is made from DNA through a process called transcription, and the RNA that is directly made from the DNA is called a nascent RNA or nascent transcript. But this is just the start of an RNA's life. Next slide, please. So, through natural biological pathways, that nascent RNA is processed, which means it's edited and revised to give rise to multiple, sometimes hundreds of different RNA molecules for each gene. For example, if you note in the drawing on the left, there are some sequences shown in tan that are successively removed in a process called splicing. And ultimately, you see that there's a mature RNA and it has 4 colors. This is only one way that nascent transcript can be spliced. If you look at the drawing on the right, you see that the 4 colors can, in theory, be spliced together in numerous ways so that the information in one gene is diversified. RNA molecules are also modified, and you can see on these RNAs the little circles, triangles, rods. These are the RNA modifications that allow further diversification. And through splicing and modification, RNA processing reverses the information encoded by DNA. And this allows the complexity that's needed for life. Next slide, please. So what exactly are RNA modifications? So let's start on the top by looking at the 4 building blocks of RNA. These are the unmodified nucleosides. And then if we look on the bottom of the slide, you'll see that each of these nucleosides, cytidine, uridine, adenosine, and guanosine, can have atoms that are added, modifications that are highlighted in gray. This slide only shows 4 of the 170 known modifications, and we think there are many more. Next slide, please. So RNA molecules are 3-dimensional, and modifications, one thing they can do is change the shape of an RNA. By changing the shape, they can alter the RNA interactions that this blue RNA here might have with the green biological molecule, maybe a protein. Next slide, please. So why do we care about RNA modifications? Well, they're crucial in nearly every life process. They're important for regulation of metabolism, circadian rhythm, immune regulation. And research has revealed that disrupting the cellular machinery that allows the modifications can lead to a wide range of human diseases, neurological disorders, heart disease, cancer, diabetes. There are so many RNA modifications linked to disease on a special kind of molecule called tRNA. That very last thing listed is that there's a phrase that's been coined, tRNA modopathies. And if you visit chapter 2, you can see a list of human diseases that are caused by aberrant tRNA modifications. If we can get a better understanding of RNA modifications and their linkage to disease, we can generate more targeted and personalized medical treatments, which in turn will advance human health. Next slide, please. So there's still a lot to explore and discover about RNA modifications, but what we already know has been leveraged to produce useful biotechnologies such as vaccines and medicines. And the most recent example of this is the vaccines that saved millions of human lives worldwide during the COVID-19 pandemic. Some of you probably received the mRNA vaccine, and if you did, on the right of this slide, you will see that the RNA vaccine, the mRNA vaccine you received, was decorated with the modification called N1-methyl pseudouridine. And that was important because it allowed your body to mount a defense against the virus, but it minimized the inflammation that could be detrimental. Some of you know that the 2023 Nobel Prize in Physiology or Medicine was awarded to the researchers whose early work studying RNA modifications for use in mRNA vaccines laid the foundation for this critical breakthrough. Next slide, please. So RNA modifications will undoubtedly continue to enable the expansion of therapeutic options. For example, mRNA vaccines against cancer are currently undergoing clinical trials. Expansion of mRNA vaccines for other pathogens, HIV, influenza, and bacteria are also possible. Understanding how bacteria and viruses use RNA modifications to evade the human immune response will also help unlock new therapies. For example, they will help in the problem of antibiotic resistance. RNA-based therapeutics that incorporate RNA modifications are already used to treat rare diseases such as spinal muscular atrophy. And if you visit chapter 2, you can see a table that lists the RNA therapeutics approved by the FDA or EMA. Next slide, please. So RNA modifications show exciting promise for enhancing agricultural productivity and pushing the boundaries of synthetic biology, nanotechnology. Preliminarily, engineering of RNA modifications has demonstrated improved crop yields and drought resistance in potatoes. And this is very exciting. And such engineering has the potential to improve food security for billions of people across the globe. Altogether, leveraging knowledge about RNA modifications is expected to greatly boost the bioeconomy. A recent report projected the bioeconomy to generate about $4 trillion per year over the next 10 to 20 years, with health and medicine and food and agriculture being major contributing sectors. The COVID-19 vaccine alone, through the reduction of excess mortality, have been estimated by the National Bureau of Economic Research to have an economic value of $3 trillion in the US. Clearly, the return on investment in RNA modification research holds immense potential. The report has a number of conclusions. One conclusion is that RNA modifications are a critical but underexplored area of research. And if you look at this list, the report notes that a more complete understanding of RNA modifications will be important for advancing fundamental knowledge of living systems, maintaining health of humans, plants, animals, and our planet, preventing and treating disease, improving crop yields, stimulating the bioeconomy, and addressing other issues of societal importance that probably we can't even imagine yet. Next slide, please. Our second conclusion is that the most impactful goal will be to enable the determination of any epitranscriptome. As you see listed on this slide, epitranscriptome means the sequences of a set of RNA molecules and their modifications. Epitranscriptomes vary between cells and tissues and with age, sex, and environment. While the committee clearly understood early on from our statement of task that our goal was to decipher the epitranscriptome, we quickly realized that our task was very different from mapping and sequencing the human genome. While every cell in an individual has the same genomic sequence, and I'll have you look to the left of this slide, we have one DNA genome, but to describe an individual requires sequencing many epitranscriptome. The epitranscriptome of a muscle cell will be very different from that of a skin cell because the processing of RNA from each gene is amazingly diverse and dynamic. We go to point 3 on the right. The bottom line is there are many important epitranscriptomes to determine, even for one individual. So our task and goal should be developing technology and infrastructure to enable the sequencing of any epitranscriptome, and this will be the most impactful goal. So if we go to the next slide, comprehensively sequencing RNA and all of its modifications is expected to have profound implications across many sectors. Yet RNA science stands at a critical crossroads with persistent gaps and technical and infrastructural hurdles to overcome. And our report identifies challenges in 5 areas, and these are listed here and treated very carefully in our report. Next slide, please. So we were also asked to generate roadmaps. And these roadmaps try to provide tangible milestones and goals. And if you want to read about these roadmaps, you go to chapter 3 and you'll see a roadmap for advancing tools and technology. And over to the right, you see that if you go to chapter 4, you'll see a roadmap for developing standards and databases. And on the next slide, I want to zoom in onto the roadmap for developing standards so you can see that we have goals for 5 years, 10 years, 15 years. And that again, these are very tangible milestones that we hope to reach. Standards are going to be huge for all aspects of this initiative. And for RNA modification researchers, we hope that at the end of the 5 years, at the end of the first 5 years, there will be a commercially available standard kit. Another conclusion shown on the next slide is that large-scale coordinated efforts in the life sciences have been vital in driving science and technology innovation. As mentioned, we carefully looked at these previous initiatives, the Human Genome Project, the Glycoscience Program, and these were successful. And we realized that such efforts hold value in their ability, as listed, to align federal agencies support public-private partnerships, organize consortia, fund individual labs, and prioritize closing gaps in technology development, standards, infrastructure build-out, workforce training, and public awareness. And of course, conclusion 3 leads to our conclusion 4 on the next slide, that a large-scale epitranscriptomic effort is needed to accelerate technological innovation and progress. And I'll note one thing on this slide. We envision that there's going to need dedicated funding to key federal entities, NSF, NIH, NIST, DOD, and DOE. And finally on this list, it goes without saying, an endeavor of this scale and scope will entail a substantial investment of time and resources. And on the next slide, I will point out there's a number of recommendations in our report. We know that we recommend that this large scale epitranscriptomics initiative will need an established oversight body such as OSTP to coordinate efforts. You'll see in some of the other recommendations, please hit the next arrow. We recommend that federal funders of research such as NIH, NSF, DOD, and DOE should invest in and prioritize closing the gaps in various areas. And we recommend that an organization such as NIST should develop, curate, and promote standards. And on the next slide, I'll just note briefly that in addition to standards, we know we're going to have management of databases and a strong workforce. And the next slide, which I think will be the last slide, we just want to note that if successful in addressing the challenges and overcoming the roadblocks in the 5 areas described in the report, we will have novel RNA sequencing tools, technologies, computational methods, data infrastructure, workforce benefits, and as I've tried to highlight during this presentation, in addition, as seen on the right, a large-scale initiative focused on epitranscriptomics will lead to a number of payoffs that we've discussed. And at this point, we can go to the last slide. And I thank you very much for your attention, and we are happy to answer your questions.
Fantastic. Thank you, Brenda. Thank you, TJ. And thank you to the rest of the committee members who have joined us here today. And I know you'll be calling on them as we work through our Q&A. So this is the time where if you have any questions, please jump into that box below the live stream where you can ask questions for our Q&A session. So I'll get us started now. So your report has a lot of recommendations, and I'm wondering if you had to prioritize them, where would you start?
I'm sure there'll be others jumping in. I would say we take the first step. To me, that's what we do. Early stages are really going to need a concentration on standards. This is going to be so important for letting different laboratories and institutions, industry, to correlate their results. And I will see if there's another committee member that wants to hop in.
It can happen. I completely agree with what Brenda said. Getting the standards established is going to be very important for ultimately enabling the technologies that we believe are going to be critical. I would also say just a very important first step is, getting this large scale initiative going. We've made a lot of progress in this area, and we hope that progress continues. But at the same time, we hope that we can really get this large coordinated effort off the ground, because I think ultimately that will be necessary to help us realize these more long-term 10, 15-year out goals of this initiative.
If I can actually say something, I think for me, the most important part is our second recommendation, that there has to be a coordinated effort to engage the federal funders and private foundations and the private sector to really, provide the ATP, the energy to get it going.
Thank you all. Our next question builds a bit on what Kate was just talking about getting started. So when you're looking at this report and looking at the field at this time, what are the immediate steps to get us started now that the report has been released? What are our next steps?
That sounds like something Jeff might be able to answer.
Well, I think that no decision is ever made until resources are assigned. Okay. And so that's both financial resources, that's intellectual resources, but it's also governance structure. And I think that the decision to move forward with this will be the trigger that we're all looking for is the assignment of resources, and where they come from will give a window, will presage the governance that's going to come with it. I think the committee feels quite strongly that this is a very large undertaking, and it's a multidisciplinary undertaking, and that we need to leverage resources across academia, across industry, but also across the many components of the government that can add to this enterprise. And I think that there's no one group that will really be able to take this and move it forward. So when we look for funding, when we look for governance, we're going to have to look for partnership as well. The place to get started is to identify resourcing, governance of that resourcing, and that will also identify accountability. And once there's resourcing and accountability, the train gets rolling.
Thank you, Jeff. And again, having a central organizing agency is going to be really important.
I also want to highlight recommendation number 6, where we recommend that various entities collaborate to produce educational materials to raise a public awareness to encourage young and motivated students to go into the field of RNA biology and RNA modifications, which will be very important to get started because generating the workforce in this field is has a long-term horizon.
Thank you, TJ. Hannah, do we have something else?
We have so many questions. Yeah, we'll keep us going. All right, next up, the report shows that there is limited research in RNA modifications. Could you all just talk a little bit more about what are some challenges and limitations associated with studying RNA and RNA modifications, and how are researchers beginning to overcome them?
Somebody in our panel that wants to take a shot at that?
Well, I will say that the history of the modification field is quite distinct in a way that initially was so hard to grasp them, that it was really hard to know how important they were. And rumor was that they were not important. Now, as detailed in this report, it is clear that some technology has been in development that allows us to map some of the modifications and provide some notion of what their function is. But it's worth pointing out that when we talk about function, we tend to talk about a function, not every function. So not being able to track them, the modifications in this case, and how can we even start to understand their full impact on cell function and organismal function? And that is the story. So what is the limitation? Currently, the technologies are excellent. No single technology addresses the problem. Many of the technologies are terribly expensive, so not accessible by a more democratized population of scientists.
Thank you, Juan. And I just would like to add that, as Juan said, we have many more RNA modifications that we need to develop the technology to detect them and the sensitivity to quantify them. But we also, the sequencing technology right now, as it stands, we can sequence usually a small region of an RNA with its modifications. But as I was talking about for splicing isoforms, if we get a small region, we don't know what's at the other end of that RNA. So we really want to focus on the goal of getting direct sequencing methods that'll take us end to end and get the RNA sequence and its modifications. It seems, I suspect out there to some of you who know about RNA modifications, that seems like a pipe dream, but that's why we've got to start now.
I'll just add, if I may, one last thing, and something Brenda brought up earlier is the importance of standards. You know, that you asked about limitations, and that's a big one right now. There are lots of really smart scientists all across the country, across the world, who have developed some really great technologies that have helped us to map these modifications and better understand them. But you know, currently the state of things is that there are a lot of different methods. A lot of them are just done in isolation for a certain modification. And you know, there's often a lack of rigorous standards that have been established, you know, to be able to benchmark these different technologies. And so I think getting that up and running for all these different modifications will be really important, not just physical standards to use in the actual techniques at the bench, but you know, computational analysis standards, nomenclature standards, all of that will be really critical.
Just add one more quick thing. An aspect that is very nicely covered by Brenda in her presentation, but also needs a little bit more discussion, is the presence of multiple modifications on RNA. Most of our current understanding and the availability of techniques enable us to study one modification at a time. But even those few modifications that were studied have given so much benefit, therapeutic benefit, for instance, in the case of COVID. But the combination of these modifications could have a lot more for us to offer in the future. The combinations could drive different cellular phenotypes, different diseases, and that is a bright future for the RNA field.
Thank you, Saurav.
Yeah, and if I can add in on top of that, Brenda, because Saurav raised a good point that we also talked about in the report, which is the need for computational tools. So not only can we take the data and understand what that data is, and as Kate said, we have standards so it's accurate, but can we get to the future of predicting what might be there? Because that will be far more empowering than have to look at every single sample over and over again.
Thank you, Pat. And no doubt, AI and machine learning will be really, really critical there, as in many aspects of our life. And Hannah, I'm wondering if there, you know, I think the collaboration between with industry, academia, industry is an important topic, and maybe I'm going to ask Nick to answer any question that Hannah might come up with. Is there anything about that?
Yeah, I'm gonna I'll pull up one of our questions that we have here. So one of the questions from our audience is, could you give examples of what you envision as the ways that public-private partnerships could work? How would those be structured? So why don't we go with that? I think that'll take us where you want, Brenda.
Thanks. Please, Nick.
Yeah, so one thing we learned from our studies of the Human Genome Project, of course, is that the biotech industry has an extraordinary capacity to accelerate technology. And for that private sector to become involved, there must be healthy competition, mutually beneficial collaboration, as this question points out. But importantly, also a sufficient market opportunity or financial incentives to get private partners involved. And so maybe just some examples of how that has worked historically. A market opportunity might be a government-funded academic lab that's seeking new technologies to carry out their research. Another might be an opportunity to develop a new RNA-related therapeutic, and the private sector would be developing technologies required to validate that therapeutic or to do quality control on that therapeutic.
Thank you very much. Anybody have anything they want to add to that?
Yeah, I might add that a construct, there are many constructs in which these sorts of discussions can occur. Okay, and they can be highly productive. One construct that is in place and active is the Manufacturing USA Network, where there are multiple institutes that focus on public-private development using both federal monies, private monies, and academic resources to move forward different technologies. And this has been shown to be highly effective. There are actually 3 biotech-related institutes, Nimble, BioFab, and the newest one, BioMade, for synthetic biology. This is certainly a construct that could be benchmarked and could be examined and maybe even leveraged rather than starting with a blank piece of paper. But there are many ways to go about this. But for the diverse stakeholders, the academic community, the industrial community, and the government community, to all be pulling their oars in the same direction at the same time, we have to have a common scorecard. And everybody has to realize that we all win and we all benefit when we're pulling the oars together. And I think there's a lot of learning in the Manufacturing USA program of things that we can improve upon, but also proven practices that can be built upon. That would be another place to look.
Thank you, Jeff. Hannah, do we have something else there?
Oh, yeah, we do. All right. Our next question is, what technology do you envision as having the greatest potential to successfully perform modified RNA characterization?
So as chapter 3 outlines and reviews very extensively, the single molecule sequencing technologies have a great potential because they can really sequence long pieces of DNA and RNA. So you can potentially sequence from one end of the RNA until the other end. And also they can also reveal at least a particular type of modification in the context of long, full-length, complex RNA sequence. There are still a lot of room to improve in terms of sensitivity and accuracy, and also ability to detect more than one modification at a time. But clearly, single molecule sequencing methods is one of the major tools that will need to be developed further.
Thank you, TJ.
Want to add one more quick thing. Thank you, TJ, for nicely pointing out the importance of single molecule sequencing technologies. In addition to the single molecule technologies, as mentioned in the report, there is a need for reagents which can amplify RNA. For instance, reagents which can make copies of RNA to make the detection of low sensitive, low abundant RNAs is also going to be an important aspect that would spur these new and emerging technologies to detect low abundant copies.
And of course, it would have to get all of the RNA modifications.
I will add, sorry, that let's not forget mass spectrometry, which gives you the highest accuracy bar none currently. But tongue in cheek, the most important technology, as perfectly outlined in the report, is a technology that currently doesn't exist. And that's why we need this major investment of resources and humans to be able to get it going and get it done.
Thank you.
Yeah. All right, we just have time for one more question. So in our final minutes here, how does this effort compare to the effort and resources needed for the Human Genome Project?
Perhaps we can ask Nick to chime in.
Yeah, absolutely. So the Human Genome Project obviously was an ambitious undertaking that we paid close attention to, as I mentioned. It was the first concerted effort of such a large scale that was applied to biology. And so it's been the precedent for many other large-scale biology initiatives that have come since, and we paid close attention to this and wanted to learn as much as we could from that. Embedded in the report are many of the lessons that we learned from this, including the importance of technology development, the thing that Juan also just pointed out, which was that they had to trust in the trajectory of innovation to come up with new technologies that didn't exist yet, and the value of a strong workforce, education, and stable infrastructure for this. With respect to the amount of resources and effort and how they may compare to the Human Genome Project, this committee didn't have expertise in terms of making financial estimates and funding estimates for what this might cost, except that we noted that the investment into the Human Genome Project paid excellent returns into the US economy and the world economy. And so any dollar spent would be put to very good use and would return massive returns to the US economy and to the world.
Thanks, Nick.
Thanks, Nick. I want to offer an opportunity for Brenda, TJ, anyone else, if there's anything in our last couple of moments here that you want to make sure you highlight as people, the report just came out a few hours ago, they might be just starting to dive into it now. Are there any things that you want to make sure people are walking away from this webinar understanding or at least looking for in the report?
I think that most of the things that we've said encompass the excitement that we feel. I really do feel this is just such a natural step from the Human Genome Project. This seems to me the most important next step. We're going to get so much information that's going to help personalize medicine. We're going to understand how we can manipulate plants, for example, as climate change starts to affect us. And yeah, I'm sure that I've drank the Kool-Aid, but it's just such an exciting step to me and one that seems so important. TJ.
Yeah, I also want to repeat what Brenda said. The report goes beyond motivation for biomedical development and health. It also touches upon food security and perhaps even clean environment. So we do research and science to improve human conditions, and how we do it while maintaining a sustainable future. And the science proposal will have a great impact on both.
Fantastic. Well, thank you all so much. Unfortunately, it looks like that's all the time that we have for questions today. Attendees, once you exit this webinar, you'll be redirected to the report page where you can find the report and all related materials. With that, I'd love to thank our fantastic committee and speakers one more time. Thank you for your time, and thank you all for participating. Have a great rest of your day.